So real quick before we start, I just wanted to pull up the Canvas page just to kind of reiterate. So I don't know if you noticed, but after last lecture, I added the recording to lecture one right here. And in general, for each week, I will be adding a link to the recordings, directly under the reflections, or not under the reflections, directly above the reflections. So yeah, if you end up missing any of the sessions for whatever reason, that's absolutely okay. And you can follow along the recordings directly from here. Another thing that I want to point out is that the week one reflection, you should have hopefully finished by yesterday. And again, the reflections, there's nothing kind of intense about them. I just wanted maybe like a paragraph, just you talking about some specific thing that you found interesting from that topic for that day. So to get a passing grade in this class, you need to submit a reflection for each week. I'm not going to be stringent on deadlines whatsoever. If you forget for whatever reason, that's totally fine as long as you fill them out before the end of the quarter. But just try to keep up with it just to, I think it's good to reflect on the stuff that you learn. Cool. So without further ado, I guess I'll jump to today's topic. Let's see. Let me share my screen and let me get the chat open. There we go. So yeah, so as always, please feel free to include any questions that you have in the chat and I'll answer them in real time. Nice, cool. So last time we talked about more broadly, what is molecular epidemiology? What are some questions that public health officials want to be able to answer? And really, what can we do as people that know maybe a little bit of molecular biology? What can we do to help track the epidemic? And a lot of the stuff that I talked about was kind of centralized on this notion of I can collect a bunch of viral genome sequences from different people that have a virus, and using that information, what questions can I answer? So today, I want to actually explore that issue. How do I collect viral sequences from patients? And today specifically, we're going to put ourselves in the shoes of the very first people that sequenced the COVID-19 genome, right? So a brand new epidemic is emerging. You see people have some unknown disease that you want to see what's causing it. This is what you would do to sequence that genome. So today's goals, we're going to talk about DNA sequencing in general. So I'm assuming, you can get by this lecture without any prior experience to DNA sequencing technologies. I'll go over everything from scratch. We'll talk about genome assembly. So how do I actually reconstruct the genome of this hidden organism? We're going to talk about some concepts called contigs and scaffolds that you'll learn more about once we get to the genome assembly section. And we'll talk about how can I assess that a given genome assembly that I've constructed is actually good. So first, DNA sequencing. So I'm going to start this with an analogy. So imagine that I have a bunch of newspapers from the New York Times on June 27th, 2000. So I have a big stack of the exact same newspaper. And then, maybe I like living my life dangerously, so I put a bunch of dynamite under that stack of newspapers. And then I light the fuse. This is, of course, completely hypothetical, so please don't do this at home. And then kapla, all of the newspapers blow up. And what's left are these tiny little fragments of a bunch of copies of the exact same newspaper. And my question is, how can I reconstruct that newspaper? How can I see what that newspaper said from all of these tiny fragments from all these different copies of that newspaper? So that's kind of the general idea that we're going to call the newspaper problem. And you'll notice in this newspaper problem, we're not just stringing together pieces next to each other. We're looking for overlaps. So this fragment and this fragment overlap in this section, and we want to try to use those overlaps to be able to piece those little fragments together. So you might be wondering, what does the newspaper problem have in common with the DNA sequencing problem? So when I'm sequencing the genome of some organism, imagine that I have multiple copies of the genome sequence of that organism. So here I have 4 copies of the exact same organism's genome. Sequencing technologies, for reasons that we won't get into here, they have a biochemical limitation that they can only basically sequence random small fragments of of that genome. So you'll notice that I kind of cut the genome, like each copy of the genome, into these different small fragments. So like I cut here, I cut here, I cut here. So I was able to sequence this fragment, maybe this fragment, this fragment. So we get these little small fragments. We can't just read it from start to end like a book. We can only read smaller fragments. And what's worse is, just like the newspaper problem, where some of those little fragments probably burnt away, we don't actually get to observe every single fragment that we generated. We only get to see a subsample of the fragments. And our question is, how can we reconstruct the genome from these tiny fragments that hopefully overlap by a decent amount? And this introduces the genome assembly problem. So the general workflow for sequencing a genome is that we start with multiple copies of the genome. We shatter that genome into a bunch of small fragments, and then we sequence those small fragments, because the sequencing technologies have limitations in how long of fragments they can read. And then using those small fragments, we want to take the overlaps of those, what we call reads. Reads are the small fragments that we sequence. We want to take their overlaps and reconstruct the original genome. So we're going to talk about what's called the DeBruyne Graph approach to genome assembly. This is a very standard approach that a lot of genome assembly tools use. So imagine that I have some hidden genome sequence that I told you in advance that these are all of the 3-letter fragments of that genome. So all of the substrings of length 3, this is that collection. And I've put them in alphabetical order just arbitrarily, because the sequencing technologies don't give us any notion of order between the reads. It's just kind of a random ordering. So I've just arbitrarily sorted them alphabetically, but that's not necessarily how they showed up in the original genome. So the DeBruyne Graph approach, we have to first construct what is called the DeBruyne Graph, which is basically a network where for each prefix and suffix of our reads, and specifically unique prefixes and suffixes, we create a node. And then we connect 2 nodes if a given read. So for any given read, we connect the prefix node to the suffix node. So for example, for this one, ACA, the prefix is AC, the suffix is CA, and the read ACA represents a link from AC to CA. Does that kind of make sense? Maybe I'll pause there if there's any questions. So basically, for each of these small fragments of the genome that I've sequenced, I take the prefix, so the first k-1 letters, where k is the length of the read, and the last k-1 letters, make nodes, and then every read defines a link from the prefix to the suffix. Kind of makes sense? Okay, it looks like it makes somewhat sense. So let's keep working through this. So the next read is ATT. So the prefix is AT. So I'm going to make a node for AT. And then the suffix is TT. So I'm going to make a node for TT. And then the read itself, ATT, represents a link from AT to TT, from the prefix to the suffix. Next one, GA. And then AT, you'll notice I already have a node. So remember, I'm making a node for each unique prefix or suffix. So I don't make another node AT, I just reuse the existing one. And then GAT is an edge, it's a link from GA to AT. TAC, TA is the prefix. I haven't seen it yet, so I create a new node. AC, I have seen, so I don't create a new node, I just reuse the existing one. And then TAC is a link from TA to AC. And then the last one, TTA, I already have TT, so I don't create a new node. I already have TA, so I don't create a new node. And then TTA is a link from TT to TA. Okay, so this network is what we call a DeBruyne graph. Okay, so now the question is, why did I even build this? How does this help me reconstruct the genome sequence of this unknown organism? Well, it turns out that we can find what's called an Eulerian path. So an Eulerian path is defined as a path that visits every single edge, every single one of these links, exactly once. So given this network, maybe take a few seconds, think, can you find a path that hits every single link exactly once? And you can only follow links in the direction that they're drawn. So like I can only traverse this link from GA to AT. So think of them as like one-way bridges. So maybe take a couple seconds. Can anyone think of, okay, so someone put, and so in the chat, make sure to set all panelists and attendees if you want to post so that everyone can see. But someone posted in the chat, GA, AT, TT, TA, AC, CA. Excellent. So let's work through this. So precisely, we can, so there is indeed one path that visits every single edge exactly once. And to reconstruct the genome, what we can do is, as we follow that path, let's start with the entire first read, so the edge that labels the first read, and then keep appending one letter at a time for each subsequent read. So the first one, GA to AT. So this link is labeled with GAT. So it's my first one. I'm going to use the whole GAT. Okay, then the next link that I follow, AT to TT, that's the next edge that I can follow, labeled by ATT. So I'm just going to take the last letter, T, and add it to the end. Right, and the next one is TTA, so I'm going to add an A. Then the next one is TAC, so I'm going to add the last letter, C. And then the last one is ACA, so I'm going to add the last letter, A. And I've actually now reconstructed my original genome sequence. So Gattaca, I don't know if you've seen it, it's a great movie. But this was actually the hidden sequence that all of these reads came from. Any questions about that? Concerns? And so my assumption here was that I had every single substring of length 3. So in reality, real data is not always this clean. There's some things that can go wrong. So for one, I assumed that I had every single substring of length 3. But in reality, some of those substrings might have disappeared, right? Just like the newspaper problem, some of them might have blown up and gotten completely obliterated. So there might be some of these short reads that are missing. And then some other problems are the sequencing machines themselves are imperfect. So even if the sequencing machine says this read is GAT, it might have some errors. So it might actually misread one of the letters of that sequence. So in practice, what people use are tools like this. This tool is called Spades. It's a genome assembler that is one of the most popular ones used. And I think it was actually one of the first ones used to sequence COVID-19. So if you're interested, here's a link to the software. You can also run it on the internet. So if you're curious about that, feel free to reach out to me. But yeah, basically the nice thing is we as just kind of users, researchers of these tools, we don't have to worry about those low-level real-world issues. Tools like Spades do all of this fancy stuff for us. Okay, so there's a question in the chat. Since the reads were arbitrarily sorted in alphabetical order, wouldn't this affect the DeBruyne graph in the final sequence? Great question. So actually, notice that the order in which the reads were given to us, it just changed the order in which we built the DeBruyne graph. So we happened to build AC and then CA and then AT and then TT, right? It changed the order in which we built it, but the graph structure itself isn't going to change. And it might not be super obvious why that is, but I can guarantee you that no matter what order you give me the reads, as long as you always give me the exact same reads, you'll always get the exact same DeBruyne graph. Yeah, great question. And the trick was we didn't necessarily look for a pattern that existed in our reads. What we looked for was in the DeBruyne graph, is there a path that visits every single link exactly once? So even if our reads are in a different order, we'll get the same exact DeBruyne graph, which means that that exact same path will exist no matter what order we read the reads in. So it's like a super clever, like, I don't know, it feels like cheat codes. Like I don't know, it's so clever and so elegant that it's so simple. Yeah, great question. So great question. So there's a question, what if there isn't such a path where we visit every edge? Edge is what we're visiting, not every node. Slight distinction, but it actually makes a big difference here. So if there's not a, so if no such path exists, that's actually a great question for the next section. So the question was, what happens if no such path exists? What if there is no path that visits every single edge? And that can actually happen. So basically, imagine that our DeBruyne graph was disconnected. And this can happen with real world data if we didn't actually observe every single portion of the genome. So if we saw this portion and this portion, but there was like a window missing, and then this portion, but there was a window missing, we won't necessarily reconstruct a single genome, but instead what we can reconstruct are smaller fragments of the genome. So imagine in this network, if we were maybe missing this link. So maybe if TAC didn't exist, we could still reconstruct GA to AT to TT, which would be GATA. Oh, sorry, and yeah, so sorry, GAAT TT to TA, which would be GATTA. But this link wouldn't exist. And we can reconstruct this piece, ACA. So spades, one of the things that it takes into account is precisely the fact that there might not exist such a convenient path that hits every single edge, in which case it'll try its best and reconstruct as long of contiguous segments as it possibly can. And those long contiguous segments are what are called contigs. So a contig is a long stretch that was able to be reconstructed. It's not necessarily the entire genome, because remember, if certain pieces of the genome just disappeared because we exploded it and we couldn't see it, we won't necessarily be able to reconstruct a single long contiguous genome sequence, but the genome assembler will output each of the longest such pieces it was able to reconstruct. And those are called contigs. And then even more so, sometimes we actually have information about the order of the contigs. So maybe we know that the first contig, the first contiguous segment, is this exact sequence. And maybe we know that the second contiguous segment is this exact sequence. And maybe we know that there's 500 letters in between them. So maybe we know that there's 500 letters in between, but we don't know what those letters are. So a scaffold is defined as an ordered series of contigs. We know this contiguous sequence came before this contiguous sequence, and that one came before this contiguous sequence. And we might actually have ideas of how much space is between them, but we just don't know these sequences that are in between. It's hidden from us. Does that kind of make sense? So what tools like spades actually output are called contigs. And our hope is that there is going to be a single long contig, but that's not always a guarantee. How do we know the possible space between contigs? That's a great question. So I'm not going to go too in depth, but basically, let's imagine that I have some genome sequence. So let's pretend this is my genome. There exist sequencing technologies that are called paired end sequencing, where we can basically reconstruct, so we can maybe sequence this part of a molecule and maybe sequence this part of a molecule. And the sequencing technology itself has information about how much space is physically between those two fragments that it sequenced. It doesn't necessarily know the exact sequence, but it roughly knows how many letters were between those two fragments it was able to sequence. And then if we have enough of those, so maybe I have another read here that overlaps with this one, and then maybe another read here that overlaps with this one, and I know roughly how much space is between them just because of the technology, then what I would be able to do is merge these two into a single contig, a single contiguous sequence, and merge these two into a single contiguous sequence because they overlap. And then I would know that the space between here and here is the space between those two contigs. So how do these sequencing technologies do this? I'm not going to go too far in depth, but feel free to look up, I'll type it in the chat, paired end sequencing. It's a really, really clever like biotechnology or bioengineering trick. So there's a question in the chat, how do we know which contigs come first? So basically like this, if we can reconstruct the contig, so maybe there's a read here and then a read here that we know the order. And we know the length of this, but we don't know what the actual sequence is. So maybe this is all one contig now. This would have contig 1, contig 2, contig 3, and we would have a rough idea of the length between them. And we would know the order of it because it's just the sequencing technology tells us that this fragment came before this fragment, and it tells us this fragment came before this fragment. So we, as the assemblers, would know this contig must have come before this contig because this fragment came before this fragment. And we would know that this contig came before this contig because we know this contig, this fragment came before this fragment. Does that kind of make sense? It gets a little weird after a while, but I can definitely, if you're interested, I can send you some resources on videos that explain these technologies. Yeah, excellent questions, by the way. This is really good. Cool. Any questions about that? Makes sense? Okay, so let's imagine that we have some DNA sequencing experiment. So we took some samples of some virus, we sequenced it, and we ran it through SPADES, which has all these sophisticated techniques for reconstructing these contigs and scaffolds. And now we have our genome assembly as a collection of contigs and scaffolds. And we want to know, how good did we do? You know, is this actually a very accurate or representative genome sequence, or is it good? Is it bad? How can we tell? So imagine that these are the contigs that we have. I've colored them as different colors, and I've just kind of sorted them from longest to shortest. So this is not necessarily the order that they appeared in the genome, right? So these are not the scaffold orderings, but I've just arbitrarily sorted them from longest first to shortest last. So I'm going to define total contig length of T to be the total length of all contigs that are of length at least T. So for example, if T was 50, total contig length 50 is the total length, the sum of the lengths of all of the contigs that are length at least 50. And I'm going to go over an example with this in a second. And one metric that's used to define how good genome assembly is called N50. So N50 is the maximal contig length such that total contig length of that length T is at least 50% of the total length of all the contigs. And this sounds really weird, but this example that I'm going to go through might hopefully make more sense. So imagine I have these 5 contigs. The longest one is length 70, then length 60, then 30, then 20, then 10. The total length, so the sum of all of these contigs lengths, is 190, just 70 plus 60 plus 30 plus 20 plus 10. So the total length is 190. And I want to see what is the N50 of this genome assembly that had these fragments. So I compute 50% of it, right? I want to know 50% of the total length of the contig. So 50% of the total length is 50% times 190, which is 95. Okay, so half the total length is 95, and I'm looking for the length, the maximal contig length, such that the sum of all contigs of that length or more is greater than or equal to 95. So let's start with the first one. So if I only look at the first contig, so the longest contig, its length is 70. So total contig length of 70 is the sum of the lengths of all contigs 70 or longer. Well, that's just 70. There's only one that has 70 or longer, which is 70. 70 is less than 50% of my total genome length. It's less than 95. So I'm not done. I have to keep going. if I consider the first two contigs, so if I consider total contig length of 60 of my second longest contig, well, that's the sum of the lengths of all contigs at least as long as 60. So 60 plus 70, because 70 is longer than 60. So 70 plus 60 is 130. 130 is greater than or equal to 95. Therefore, the N50 of this genome assembly is 60. It's the length of the longest contig such that if you add up all the contig lengths greater than or equal to that length, you get more than half the total length. Does that kind of make sense? This is a little weird, but any questions about how I got that? So basically, I first computed the total length, then computed half the total length. Then I kept trying to figure out what is the largest possible contig length such that if I added up all the contigs that length or more, I would end up with greater than or equal to half the contig length. Okay, so just to make sure that this kind of makes sense, here's maybe a hypothetical question, and feel free to post your answer in the chat. Hypothetically, if I'm doing a genome assembly, is it desirable for the N50 to be bigger or smaller? So do I want this number to be bigger or do I want it to be smaller? Maybe think about it for a little bit, and as you have ideas, feel free to type them in the chat. If you can, select all panelists and attendees, just so that everyone can see your response, if you're comfortable with it. So my question is it better for the N50 to be bigger or smaller? Feel free to take a little bit of time thinking about it. So I'll read out some of the answers in the chat. So one of the answers is a bigger N50 would be better because it indicates you have more long contigs. Someone else said bigger N50 is probably better. Bigger because it means your contigs are bigger. Precisely. So let's step back and think about the best case scenario. So the best case scenario is if I was able to just, in a single run, sequence, that's kind of hard to read. Let me do purple. So the best case scenario is if I was able to just sequence the entire genome in one single contig, right? That's the absolute best I can do. Anything split up is just a matter of I didn't have enough of the reads that sequenced it, which split up my contigs, because my DeBruyne graph had the pieces missing from it. So the best case scenario is if I have some viral genome, I want to just reconstruct that entire thing in one long sequence. So what would be, so in this example, that long sequence would be the entire length of the genome, and the N50 would be the entire length of the genome, right? It would be the length of that single super long contig. So it's a little weird to think about, but yeah, because the best case scenario is that I want to reconstruct the whole genome sequence in a single contiguous sequence, and that would result in the biggest possible N50, then that would be the best possible scenario. Bigger is better. There's a question in the chat, longer contigs equals a more complete sequence. Yes, right? Because there's only one lengthy genome for a virus. A viral genome is just one long contiguous sequence in reality. So any fragmentation that I get is purely because I was unable to reconstruct that entire long sequence. I was only able to maybe reconstruct this portion and this portion and this portion. So any shortening that I do is a failure on my part. It's me not being able to reconstruct certain segments of the genome. Any questions about that? It's a very weird thing that I'm throwing at you. So if it's kind of confusing or if you have to think about it, that's totally okay. It took me a while to wrap my head around this as well. Okay, so it seems like it makes sense. So right off the bat, I've introduced a metric, N50, that if you used spades or some genome assembly tool to reconstruct a genome, to assemble a genome, given your genome assembly, you can compute the N50, and the bigger your N50 is, the better. So that's one metric for potentially determining how good your assembly is. Okay, so the previous example, the N50, was only dependent on my genome assembly, right? If you gave me my contigs, I was able to compute the N50 purely from my contigs. But what if I was missing a huge fragment of the genome? My N50 could be as high as possible, right? I could have a single contiguous sequence, but it might not be the whole genome. It might be some tiny fragment. So there's a slight extension of the N50 metric called the NG50 metric, where the G stands for genome length. And this is basically, imagine that we're not the very first people sequencing it, or maybe using some biochemical techniques, we somehow know in advance roughly how long the genome length is. So for COVID-19, the genome length is 29,000. If I have some sample, I can determine roughly what the length is using just biochemical techniques. And then in advance, maybe I know that I'm expecting maybe roughly 29,000 letters. So the NG50 is very similar to the N50, but instead of using the total contig length, I'm using the total genome length that I was given in advance. So in this example, instead of saying a total contig length of 190, let's imagine we knew in advance that the genome length is actually roughly 270, but same exact contig. So I've reconstructed the exact same genome in these small fragments of length 70, 60, 30, 20, and 10, but I know that the true genome length is 270. So the NG50 length is the maximum contig length, such that total contig length of that is at least half the total genome length. So same exact thing, just using genome length instead of total contig length. So first thing I have to do is compute what is 50% of my total genome length. 270 times 50% is 135. So let's start where we were last time. So last time we considered the first contig, its length of 70. So total contig length of 70 is 70. That's less than 135. The second contig is length 60, like second longest. So total contig length of 60 is 70 plus 60, which is 130. That's less than 135. So I'm still not done. I still haven't reached at least 50% of my total genome length, which is 135. So now let me consider the next longest contig. So the next longest contig is 30. So total contig length of 30 is 70 plus 60 plus 30, right? That contig plus everything longer than it, which is 160, which is greater than or equal to half the genome length, right? It's greater than or equal to 135. So this tells me that the NG50 is 30. So NG50 is a slightly more accurate metric than the N50 that we can use if we know the true genome length. So Anish asked, just to clarify, in N50, we don't know the whole genome length for sure. We only work with a portion of the genome. So with N50, you may or may not know the total genome length, right? With N50, the N50 metric doesn't depend on the true genome length, right? So even if I knew the true genome length, I could still compute N50 if I wanted to by just looking at my contigs. But NG50 does require the genome length. So if I do know the genome length in advance, I can compute NG50 and I can compute N50. Both metrics are totally fine. It's just that NG50 is a slightly more accurate metric. The reason why N50 uses the total contig length is because it's supposed to be a safe metric for even in scenarios that we don't know the true genome length in advance, right? So it's less meaningful because our total contig, like all the contigs that we sequence, might just be missing parts of the genome that, the N50 wouldn't be able to take that into account. But sometimes that's the best you can do. Versus NG50 is like a superior metric that now takes into account this additional information. So they're not opposites. It's just NG50 is the better one to use if you have this information available to you. Does that kind of make sense? Yeah, no problem. Yeah, great question. Cool. So another thing that we can do is create what's called a contig length plot. So there's a tool called QUAST, which is, I think it stands for Quality Assessment Tool or something like that. This is a tool created by the exact same lab that created SPADES, but it's used to do quality assessment of a genome assembly. So these metrics like N50 and G50, you as the researcher would never have to compute them by hand. You would take your genome assembly that you got from SPADES and just feed it to a program like QUAST, and QUAST would compute all these metrics for you. And also what's really nice is it'll compute a plot like this. So basically this genome contig length plot, what we're doing is, again, sorting the contigs in decreasing order of length, and then just kind of like pasting them on this plot where the horizontal axis is the percent of the total length. So the entire x-axis is 100%. But this sequence, the longest one, is what, like 37% or something like that of the total contig length. Then this one is some portion of the total contig length. But basically, we're plotting them where the y value is the length of that contig. So this contig, the longest one, is length 70. And it's this portion of the total contig length. The next longest contig is length 60, and it is this portion of the total contig length. And you keep going, and keep going. So again, I want to just take a step back and maybe think about this. So what would be desirable in this plot? What would be a quote unquote good quality genome assembly contig length plot? Like, how would we want this to look in the ideal world? Maybe go ahead and think about it for a little bit and feel free to type in the chat. What do you think would be a good genome assembly's contig length plot? What would it look like? Yeah, so someone mentioned in the chat, longer horizontal lines, less downward jumps. Someone said straight line. Any other thoughts? Straight horizontal line at the top. Yeah, exactly. So you're all right on the money. So basically, again, let's step back and think about the best case scenario. The best case scenario is that I can sequence the entire genome in one long chunk. If I were to do that, then there would be a single contig whose length is the entire length of my total contig length, right? So the very, very, very best scenario that I can have is a single horizontal line at the top. Any deviation from this is bad, right? So in general, more area under this curve is better. Does that kind of make sense? Right, so the more the area under this curve, the better. And you can think about it because also if we think of the flip side, if we think of the worst possible scenario, imagine I have a bunch of these tiny fragments that I wasn't able to piece together. The area under this is super, super, super tiny. Does that kind of make sense? So you're all right on the money. That's perfect. Cool. Okay, so let's imagine I construct my genome assembly, and then I want to see the quality of it, which I do based on this, right? I run QUAST, I get the N50 metric, G50 metric. I get this fancy plot where I can look at it and see, like, what is the area under this curve? How long of a straight line is it? All these types of things. So I look at this and I'm like, okay, maybe it's a pretty good genome assembly. So the next question that you should be thinking as a scientist kind of on the front lines is, well, what the heck is this virus, right? I've reconstructed the sequence, but what does the sequence mean? Like, what is actually in this sample that I've collected? So there's another tool called BLAST, and this is kind of like the Google search of bioinformatics, where you basically can type a sequence into BLAST and click search, and it's going to scan this big database of already known sequences and tell you what your sequence looked like. So here on this slide, I have a figure that this is basically just a screenshot of what the BLAST results look like. So I took the original first sequenced COVID-19 genome, and I restricted my BLAST search to only include sequences that existed back in November 2019. So back before the COVID-19 pandemic even started. And just to kind of put myself in the shoes of the scientists that first sequenced the COVID-19 genome. And you see, very interestingly, all of the matches are basically to bat SARS-like coronavirus, SARS coronavirus, so, right? Everything is related to SARS or is something that's SARS-like. And the most similar one is this bat SARS-like coronavirus. So this is how scientists can figure out, okay, what type of virus are we dealing with? What properties does it have? What does it look like? And over here, we can see a bunch of metrics of like, so query cover is what percentage of our sequence that we searched for. So in our case, our viral genome, what percentage of our viral genome matched that database entry? So this tells us 95% of our viral genome that we just sequenced matched this bat SARS-like coronavirus. And as you go to like SARS, it goes down to like 88%. It tells you the score that you see here, it doesn't really mean much. But percent identity is how much of it is actually completely identical. So 89% of it was was completely identical to this bat SARS-like. And yeah, this kind of it's usually the first step as a researcher that you want to do to see what is in this weird sample that I have. Any questions about that? Any concerns? So there's a question in the chat. What happens when it is a new genome, such as when coronavirus was first found and there wouldn't be these entries? So actually these are only results that existed before COVID-19. So when it says SARS coronavirus, this is talking about original SARS, like SARS from 2008. So we're actually matching existing SARS coronavirus sequences. And even if, let's even step further back and put ourselves in the shoes of the 2008 scientists that were investigating SARS pandemic. If they sequenced it, sure, these results wouldn't exist, but there would be other coronaviruses. So coronavirus is kind of an umbrella of the viral world. It's a type of virus. So basically, even if we wouldn't get super, super precise similar matches, we would just look for what is the most closely related thing that exists. So kind of what's interesting to us is what is it and how similar am I to that? Right? And if we didn't have anything remotely like this, then the closest matches would have a much lower percent identity, much lower identical match. So there's a question in the chat, using this database, are you able to track the mutations that occur? You actually, so you can kind of see what mutations, not entirely. So later on in this course, I'm going to talk about how we can keep track of the mutations that occur as well. So you would be able to match something that has the most similar mutations as your specific sequence. So like, for example, if today I took a COVID-19 sequence and I ran it against BLAST with today's database, which now includes many COVID-19 sequences, the ones that it would match most closely to are the ones that have the same mutations as that sequence that I obtained. But there's actually better ways that researchers can kind of keep track of the spread of mutations across the population. And I'll go over that in future weeks. But that's a great question. Any other questions about this? Feel free to like type it out. So that's all my slides, but kind of the TLDR of today is basically when you're the first person sequencing some brand new, I'm just going to say pathogen. We don't even necessarily know if it's a virus or a bacteria. There's some pathogen that's causing some disease, and you want to know what is this pathogen. Usually the first thing that modern scientists do is collect a sample sequence it. Let me see how good my, then do a genome assembly. Let me see how good my genome assembly is. And then search on BLAST to see what other things have already been seen that look similar to this, just to kind of get an idea of what type of pathogen this actually is. Cool. Any other questions? Nice. Okay, well, that's all I had for today. So if you have any other questions throughout the week, please feel free to post on the discussion board. If you post on there and I don't see it, feel free to email me just to kind of ping me. I don't think Canvas sends me notifications. So yeah, so if you post something on the Canvas discussion board and I don't reply to it, feel free to email me and I'll take a look. But yeah, thanks everyone for coming. I look forward to seeing you all next week. Cool, thanks everyone. Bye everybody. Happy Monday.